So you have to remember the nitrogen fixing bacteria, what kind of the bacteria they are. So you will get 4 options, that which of the following is the symbiotic bacteria fix the nitrogen and all that. So now the next part, this is also very important. This is the nodule formation. So actually, all the pulses plant, they have nodules in their root, right? Nodules, small nodules. The you can see in this diagram. So all the pulses plant, they have nodule-like structure. All the pulses plant. So pulses comes under a subfamily a family Malvaceae, right? So the plant which belong to Malvaceae, like gram, like pea, and all that, they all have nodules in their root, and these nodules are basically meant for the nitrogen fixes. So how does these nodule fix the nitrogen? Okay, look, this is the root system with the root nodule. Now what happened? First, these are the root hair. Root hair covered by free living rhizobia bacteria. So there is a bacteria, right? That bacteria cover around the root hair of this plant, right? Root hair of this plant. Basically, the name of the bacteria is rhizobia. So the full name of bacteria is free living. So what you have to remember, first thing, this is the free living bacteria. Means it's not a symbiotic bacteria, right? First thing, this is the free living bacteria. Another thing, that it's a name of rhizobia. So actually, its complete name is Rhizobium leguminosarum. What is the name? Rhizobium leguminosarum. Leguminosarum. And we call them rhizobia. So these rhizobia bacteria, they come around, they cover the root hair. After that, what happened? That is important. This is the question. So these bacteria, they release a kind of the chemical, and the name of that chemical is a nod factor. Nod factor, this is written over here. The name of that chemical which is released by the bacterial body is nod factor. That nod factor causes the curling of root hair, right? So curling of root hair causes. This is the first question, right? So which component causes the curling of root hair? That is a nod factor. And they erode the cell wall, means they destroy the cell wall. When they destroy the cell wall, they penetrate inside the, like this way. So they have destroyed the cell wall. Formation of infection thread takes place through which rhizobia enter into the root cell. What happened? They have eroded the cell wall of the root hair. When the cell wall of root hair has been eroded, they enter inside and they make a thread like a structure. This is called infection thread, right? So they make infection thread. Then what happens? Infection thread reaches to the adjacent cell. When we talk about the adjacent cell, basically this infection thread reaches to the cortex cell, basically cortex cell. And what kind of the cell are? Basically, cortex is made up of parenchymatous cell, right? Parenchyma, right? Parenchymatous cell. Okay. When it reaches to cortex, the root cortical cells, right? They produces nodule. So what happens? They start producing formation of the nodule, right? Formation of root nodule formation start, right? And cortical cell start secrete auxin and nodule formation. So let me write it. Let me write more clearly so that you could get the writing actually. Cortical cell means the cells of the cortex. The cells of cortex after the infection, what happened? Cells of cortex multiply, comma, produces auxin, So when auxin production takes place and formation of nodule takes place. Formation of nodule takes place. So you have to remember the auxin, you have to remember the curling of the root, this nod factor curling of the root, and name of the bacteria Rhizobia leguminosa. Is that clear? I think it should be clear to all of you. Clear?
Yes, sir.
Very good. Then next one. This is very important. This is also very important. And you must have gone through with it. Look, there is an enzyme which is found in these bacteria. And the name of this enzyme is nitrogenase enzyme, right? So nitrogenase enzyme, what is the function of nitrogen enzyme? It help in fixing atmospheric nitrogen in bacteria. Nitrogen in bacteria. What is the most important thing for this one? That this nitrogenate is this enzyme, it do not work in aerobic condition, right? If oxygen is present, so in presence of oxygen, this enzyme is active. Let me write it down. In presence of O2, this enzyme is active. So can anybody tell me, in presence of oxygen, this enzyme is inactive? I'm sorry, this enzyme is inactive, right? So when it will become active, it will become active in the absence of oxygen. It will become active in absence of oxygen. That is anaerobic. Environment. During the nitrogen fixation, which provide the anaerobic conditions? Please tell me. Is there an intelligent person who is going to tell me which component provide anaerobic environment to this enzyme? Let me give you a hint. That component is called oxygen scavenger. Any one of you? The nodules, one more important hint, the nodules which are found in the legume plant, they are pink in color, and that thing provide pink color, impart pink color to this one, to these nodules. Nobody?
Sir,
yes.
Hemoglobin is like
very good. Yeah. What is the name of that hemoglobin? Very good. Excellent. Yes, Heather.
Sir, I don't remember the exact name of it, but
okay, very good. Excellent. Good, Adam. Look, actually, what happened? There is an enzyme which is found in these nodules, right? The name of that enzyme is nitrogenase enzyme, right? But as I have told you, this is please try to understand, nitrogenase enzyme. Actually, this nitrogenase enzyme, it will work only in anaerobic condition. I have written it over here, right? So, which will provide, basically oxygen is present over here. So actually what happened, there is a pink color component which provide pink color to nodules, right? So look, this is the root nodule, this is the root nodule, and there is a pink color component. There is a pink component or pink compound, found in nodule. What is the name of that pink compound? Basically, the name of that pink compound is leg hemoglobin. Leg hemoglobin. Leg hemoglobin, what is do? It make a bond with, please try to understand, you will get the question from here, you can get the question from here. Leg hemoglobin. So this leg hemoglobin is found in these nodules, right? It is a nodule. So inside the nodule, there is a leg hemoglobin. This leg hemoglobin, it make bond with oxygen. Make bond with oxygen and create anaerobic environment, right? Anaerobic environment. So due to presence of anaerobic environment, right? So due to presence of this leg hemoglobin, this nitrogenase enzyme get anaerobic environment, right? And this leg hemoglobin, it impart pink color, right? Impart it. This leg hemoglobin is also known as, it is also what we call it, also called oxygen scavenger. Oxygen scavenger. Scavenger. So that you need to remember. This is called oxygen scavenger. This oxygen scavenger. So look. So what happened? This enzyme, nitrogenase enzyme, it become active in absence of oxygen. Leg hemoglobin provided an anaerobic environment. Enzyme combined with substrate nitrogen. So it take the nitrogen from the environment and make a bond with nitrogen. Then what happened? Two hydrogen come and reduction reaction takes place. Again, second time reduction takes place, two more hydrogen come. Again, third time reduction takes place, two more hydrogen come. And then what happened? The formation of this enzyme gets separate and formation of two ammonia molecule takes place, right? That is a function of nitrogenase enzyme. That's how the nitrogenase function. But provided the nitrogenase is become active in an anaerobic environment, that thing you have to remember. And anaerobic environment is provided by leg hemoglobin. Leg hemoglobin is like the hemoglobin which is found in our RBC. It is the same thing, like same composition, and it provide pink color to the root nodule. Is that clear or not? It's very, very important part. Clear to all of you? Sania, clear?
Sir, leg hemoglobin is found in the nitrogenous enzyme.
Leg hemoglobin is found in this one, nodules, inside nodules. And it is produced by the cortical cells. Arpit.
Okay, sir.
So clear, guys? Sania, is that clear? Iram?
Yes, sir.
Very good. Anna, clear? It's very important. That's why I'm asking.
Yes.
Fine. So, very good. If you get this concept. And remember all the point, you have to remember only those points which I have written on the screen, right? Now there is
Sir, I
another
have a
reaction.
doubt, sir.
Okay, fine.
Sir, after the release of NH3 in the final stage, sir,
Right.
can this left enzyme be used again to produce
Yeah,
another-
enzymes are always reusable. Only hormones are not reusable. Enzymes
Okay, sir.
always get reused, right? Hormones do not get reused. Fine.
Okay, sir. Yes, sir. Okay.
Taken the screenshot?
Yes, sir.
Great. Now there is one more reaction that is called ammonification. It's not that much important, but still. Look, how does this ammonia get fixed, right? So look, ammonification is the process that act upon nitrogen excretion, production of the dead body of the living, right? There are ammonification. So what they fix the, or they convert the ammonification means they convert the dead part of the living organism, their dead part, from their dead part they produce the ammonia, right? So there are few bacteria, Bacillus vulgaris and actinomycetes, right? So Bacillus vulgaris and actinomycetes are two bacteria. They act upon nitrogen excretion, means like urine, right? Excretory part, and production of and they from the dead body they produces ammonia. So look, protein is the component of either plant or animal, right? So all the living organisms they have protein. These bacteria break down protein into amino acid. Then what do? So this is the formula of generic formula RNH2. This is the generic formula of amino acid. Then what happened? They break down into ROH, means organic acid, and into ammonia. Then further ammonia get reduced and formation of ammonium takes place. The process is called ammonification. What you have to remember? You have to remember only the name of this bacteria, these two bacteria. This is important for your medical. Next thing is nitrification. So what is the nitrification? Nitrification means formation of nitrate. How does the formation of nitrate takes place? Look, there are certain bacteria, Nitrobacter, Nitrococcus, Nitrosomonas. So they are responsible for nitrification. This ammonia, what happened? This ammonia combined with the oxygen, react with oxygen, and formation of nitrite takes place. Nitrite, hydrogen and water and energy is produced. So they combine what these bacteria do. They combine the inside the reaction goes on. They, with the help of enzyme, they combine the ammonia with the, means they oxidize the ammonia, and formation of nitrate takes place. After that, nitrate is again oxidized, and uh, uh, formation of nitrite takes place, right? And energy is produced. So this process is called nitrification. Means formation of nitrite is called nitrification. What do you have to remember? You have to remember the nitrification bacteria: Nitrobacter, Nitrococcus, and Nitrosomonas. So in both of the cases, what you have to remember, that is only the name of the bacteria, which is responsible for nitrification, which is responsible for ammonification, right? Synthesis of amino acid is not that much important, but still it's part of syllabus, so I'm explaining you. How does the synthesis of amino acid takes place in the body of uh, plant, right? How does the plant produce amino acid? Because we do not have capability of producing amino acid. We get the amino acid, or means we get the protein from the body of the plant or animals, right? So finally, we get the amino acid from the body of the animal plant, right? So uh, synthesis of following, there is a following method: reductive amination. In reductive amination, ammonia combined with keto organic acid and keto organic acid. For example, there is a alpha ketoglutaric acid and oxaloacetic acid, right? So these are the example of keto organic. So ammonia combined with keto oxalic acid and formation of amino acid takes place. This process is called reductive amination, right? The example is given below. These are the example. For example, alpha ketoglutaric acid combined with ammonium and NADPH, means the energy is required. Dehydrogenase enzyme work in the presence of dehydrogenase enzyme. They convert glutamate, water, and NADH. Glutamate is amino acid. Again, oxaloacetic acid, this one, oxaloacetic acid, it combined with the ammonium, NADPH is required, means energy is required equal to 3 ATP. They produces the aspartate, and aspartate is an amino acid, right? Water and NAD plus. So this process is called reductive amination. You have to remember only this part, this part, this part. The another process is called transamination, and there are only two methods of production of the amino acid in the plant. In transamination, what happened? This amino group is transferred from alkyl group to another component. So here, this is the amino group. This is alkyl group having the amino group. This amino group is transferred to keto group, right? Then what is happening? For example, glutamic acid is there. Glutamic acid have this amino group, and there is a oxaloacetic acid, right? So this is a keto group. So glutamic acid, it gives its amino acid to it gets its amino group to oxaloacetic acid. Then formation of alpha ketoglutaric acid and aspartic acid takes place. Aspartic acid is a type of what? It's a type of amino acid. So there are two processes: transamination. In transamination, transfer of amino group takes place, and in reductive amination, the reduction of particular acid takes place, so that the formation of amino acid takes place. Clear?
Yes, sir.
You have to remember only the process. What is the reductive amination? What is the transamination? That's it. Need not to remember these reactions. So this was all about the mineral nutrition chapter. Go through with this chapter and ask me if you have any doubt. You have next 2 minutes. Go through with the chapter and ask me if you have any doubt. Then I will start with the next chapter. Just quick go through with your NCRT and ask me if you have any doubt, any question.
Sir, what is critical concentration?
What is critical?
Concentration.
Critical concentration. Look, the critical concentration is that concentration beyond which, right, a particular, like it belongs to toxicity. So critical concentration is that concentration beyond which the particular element becomes the toxic.
Okay.
Was you there when I was telling you the toxicity? Look here.
Oh, sir, actually, I was.
Toxicity of micronutrient, right? Suppose that a particular concentration beyond which a tissue loses 10% of its organic weight, 10% of dry weight. It means this is considered toxic. So beyond the critical level, any of the mineral element become the toxic for the any plant.
Okay, sir.
Anyone else? No? Okay, fine. So don't get panic from this chapter. Whatever I have told you, right? You will definitely you might have got the link of this these PPT. So macronutrient, micronutrient you need to remember, and for this one, right? I have already told you yesterday that you have to remember only those things where I have made double tick and stop and this one encircled or made the star, right? So only you have to remember only those things, not any other thing, right? You have to focus over there only, right? I have only because to cram it whole list is not easy, right? And you don't have time for that, right? So when you have limited time, then this is the time to work smartly, right? And a smart work always pay off. Fine. Toxicity, I told you that this is very important. Nitrogen cycle, it's okay. Nodule formation is very important, and this part is very, very important, right? And then the rest of the part are okay. Now the next chapter which we are going to study, that is the photosynthesis. We are going to cover this part, photosynthesis. Just right. So before starting the chapter, there are some factual things, and these factual things have been asked as a question. So that's why we are going to discuss that. Look. Okay, fine. So let's start with the photosynthesis. So there are some factual part. So a question, there is a very old question. So old question of medical examination. Before starting, I want to ask a question. What is this? Is the reaction in front of you, right? In front, 6 CO2 means 6 molecule of carbon dioxide. We are studying this reaction since our childhood, like from the 9th class onward, even before 6th class, right? 6 CO2, 12 H2O, C6H12O6, 6 H2O, and 6 CO2. I have a question. What is the waste product in this reaction? Which component is the waste product in this reaction? Who will tell me?
Oxygen.
Oxygen.
Oxygen. Okay, here, more people in the side of oxygen. No one. What is Avino? Yes. Yes, Martin.
Water.
Which one? Pardon, please. Marita?
Water.
Water. Excellent. Right. Oxygen is a byproduct. I have written over here. Oxygen is a byproduct. Water is the waste product. Actually, these 12 molecules of the water have been utilized. No, sorry, not utilized. 12 molecules of water are participating. Out of them, only 6 are utilized for the formation of what? For the formation of oxygen, and rest of the 6 goes in waste. So water is the waste product. So what is the oxygen? Oxygen is a byproduct. And what is the product? Glucose is the product. Remember, right? So 6 water molecule goes waste during the photosynthesis reaction. Remember that thing. 70 to 80 billion ton of carbon dioxide is fixed annually, means every year, by the photoautotroph. All the photosynthetic organisms, they perform photosynthesis, and 70 to 80 billion ton of carbon dioxide is fixed. 170 billion, again, 170 billion ton of dry organic matter is produced annually every year. According to older estimate, this is important, this part is important. According to some older estimate, 10% are produced by land plant and 90% are produced by aquatic plant. This is an older estimate. You will get this data in the older books. But according to the present estimate, 68% of total dry weight is produced by the land. Right? That you need to remember. Again, there was a scientist, his name was Joseph Priestley. All these scientists have been asked already. You'll get this question in your previous year question paper. So Joseph Priestley was the person, and basically the Joseph Priestley was the same person we have discussed before. Joseph Priestley was the same person. Okay, Hannah have also told that water is a waste product. So Joseph Priestley is basically the same person who have discovered the oxygen, right? He is the person who have discovered the oxygen. This is the same person who have discovered the oxygen, right? So what happened? Joseph Priestley said that plant have capability. He taken a mint plant, and he said that plant have capability to convert a foul air into pure air, right? So plant have capability to convert the impure air into pure air. Now, what he given the term for impure and pure air? Joseph Priestley said that foul air, he given a term. For foul air, he given a term phlogiston, and for pure air, he given the term dephlogiston. Will you remember that? For foul air, he given the term phlogiston. Dephlogiston have been asked. What is the dephlogiston? So dephlogiston. So Joseph Priestley said that plant mint. He taken the experimental organism mint plant. He said that mint plant have capability to convert phlogiston into dephlogiston. Dephlogiston is the pure air, and phlogiston is the. So what is the dephlogiston? Now Lavoisier was another scientist. Lavoisier, he was basically he was a chemistry scientist. So Lavoisier, he said that phlogiston is a carbon dioxide and dephlogiston is oxygen. That was proved by Lavoisier. That thing you have to remember, right? Then there was a scientist. His name was the von Meyer, right? Von Meyer said that the green plant, they purify the air. Then another scientist, Reuben, Hasid, and Kamen. So Reuben, Hasid, and Kamen, they use the O2. They use the O2 and heavy isotope of the oxygen. That is the O18. O16 is the light isotope, normal isotope, and O18 is the heavy isotope. They proved that oxygen comes from water, not from the carbon dioxide. That's what they proved. Clear? Another scientist, Calvin Melvin. So Calvin he told a Calvin pathway. And Calvin said that carbon dioxide fixation, how the carbon dioxide fixation is done, right? He given the Calvin cycle, right? Or this is also known as the C3 cycle. So C3 cycle or Calvin cycle was given by Calvin, and he used the carbon 14. It's a radioactive component. He used a carbon 14 for to prove that, and he got the Nobel Prize for the same. Calvin. C3 cycle or the process of carbon dioxide fixation, right? Let me write it down. The process of CO2 fixation was explained by Calvin. And that's why Calvin Melvin, there were two scientists basically, Calvin Melvin, they got the Nobel Prize for the same. Hill and Pendle, yes.
Can I explain the before the Kelvin, the Rubin, yeah, the Rubin acid and Kelvin part again?
Which one? Reuben Hasid and Kamen. This
Yes,
one.
sir.
Reuben Hasid Kamen, they told that the oxygen comes out from the water, not carbon dioxide, not from the carbon dioxide. Right? There are three scientists. And then Calvin Melvin explained how the fixation of carbon dioxide is done. And to prove that water, this oxygen comes from the water, not from the carbon dioxide, because both have the oxygen molecule, right? They use the heavy isotope of oxygen to trace that, right? Hill and Bandel, they propose the Z scheme. We will discuss Z scheme in this chapter. Peter Michel, he given the chemoosmotic theory of ADP synthesis. This question have been asked. Who given? Michel. Michel given the chemoosmotic theory, right? Chemoosmotic theory explain how does the formation of ADP takes place, right? Hatch and Slack, they discovered the C4 pathway. We'll discuss that pathway. One more important thing, the spectrum of a visible light is 390 to 760 nanometer or 3900 angstrom to 7600 angstrom wavelength. And this is important for, and this is usable for the photosynthesis process. And PAR is photosynthetically active radiation. Photosynthetically active radiation. Active radiation. So basically, the photosynthetically active radiation, like the radiation which is responsible for photosynthesis, where the range for is from the 400 to 700 nanometer, right? So I think these were some factual point, and it should be clear to you, right? Now let's discuss with the main topic, right? Come to
So
the main.
we have to remember all the scientists. now.
Exactly, you have to remember all these things because I have incorporated only those things which have been asked. PAR already asked. Let me tell you, PAR already asked. Chemoosmotic theory already asked, right? And then this one, this Rubin Cameron already asked, and this one, Deflogiston already asked, right? And which one? This one, the amount of the dry matter 170 billion. This question I have already asked.
Okay.
And this waste product and byproduct, this is very old question. You won't get that much easy question. Now, where does the photosynthesis takes place? Look, I will discuss this structure of the chloroplast once more in the cell biology as well. Here we will discuss only those aspects which is important for the photosynthesis, right? These are the chloroplast. Okay. Now, where the chloroplast are found? So basically, leaves have a particular kind of the cell. Can you tell me where the chloroplasts are found? Chloroplasts are found in the leaves. That we all know.
So what was your question?
Where does this chloroplast is found? Sorry, which cells are responsible for photosynthesis, or where does chloroplast is found? In which cell? Leaves have a special
So
kind of cell.
mesophyll cells.
Grana.
Very good, excellent. Mesophyll cells. Right, very good. What is, and now this was not the question, but question is that the NEET question which I've been asked. What is the orientation of this chloroplast in mesophyll cell? So these leaves have a particular kind of the cell which are responsible for photosynthesis. Look, they are the mesophyll cell. So leaves have mesophyll cells. Mesophyll cells are basically meant for photosynthesis. Meant for photosynthesis. Right? Mesophyll cells are meant for photosynthesis. Inside the mesophyll cell, basically the chloroplasts are found at the peripheral region, so that they could get the maximum amount of the light, and photosynthesis could be performed. And they get the light. So these are the chloroplasts. And chloroplasts are basically found in the peripheral part. That question I have been asked. So basically, the mesophyll cells are meant for in the leaves, especially mesophyll cells. They have been designed to for photosynthesis. And inside the mesophyll cell, these are the chloroplasts which are found in the peripheral region, and these chloroplasts are meant for photosynthesis. So now come to the structure of chloroplast. This is the structure of chloroplast. Chloroplast is double membrane structure or triple membrane structure or single membrane structure.
Double membrane.
Right. This is basically double membrane structure, but yet, yes, some scientists say this is triple membrane structure. Some scientists considered that these grana are the third membrane. Right. Okay. There is an outer membrane, and inner membrane. And there is a space between the outer and inner membrane that is called intermembrane space. Inside this chloroplast, there is a gel-like substance that is called stroma. Right. And in this gel-like substance, there are these kind of the structure. These structure are known as granum. So singular is granum and grana is plural, right? So this one stalk is called granum, right? And these granum are made up of these kind of the structure, coin-like structure. These coins are known as thylakoid, right? So grana, the surface of the grana, on the surface of grana, there are certain photopigments, right? Means chlorophyll molecule, right? And in the granum, the water breakdown takes place. And this is called photolysis. Please try to understand what I am saying. In the granum part, photolysis of water takes place. What is photolysis? Breakdown. Photo means light. Lysis means breakdown of water molecule. So breakdown of water molecule in presence of light is called photolysis. So in granum membrane, breakdown of water molecule takes place. When water molecule will break down, formation of 2 hydrogen ion, 2 electron, and oxygen molecule will take place. So breakdown of the water or photolysis of water is called light reaction. We divide photosynthesis in 2 parts, light reaction and dark reaction. So what is the light reaction? Light reaction is also known as, what do we call this? Light reaction is also known as Hill reaction, right? This is also known as Hill reaction, right? So light reaction or Hill reaction is the reaction where breakdown of water molecule takes place and formation of oxygen takes place. Now what is happening inside? So this is the grana, and this gel-like substance called stroma. Stroma is the part where fixation of carbon dioxide takes place and the Calvin cycle goes on, right? This is called dark reaction, right? So light reaction and dark reaction, they collectively they are called as the photosynthesis. Light reaction takes place only in daytime. Light reaction takes place only in daytime, day only. But dark reaction do not require any kind of the light. So it keep going on day and night both. Day and night both. Right. Again. Now there is some more technicality. These thylakoids, so I have told you that this coin-like structure are known as thylakoid. The thylakoid, where the thylakoid are in contact with each other, these part are called appraised part, like this one. These part are called appraised part. And where thylakoids are not in contact with each other, they are called non-appraised part. So appraised part have appraised part, these appraised part, this one. These appraised part, these appraised part. These appraised part, they have photosystem second. What is the photosystem? Photosystem is a type of chlorophyll molecule. Chlorophyll molecule. We'll discuss photosystem. So photosystem second. Photosystem second is responsible for photolysis of water, right? Is that clear or not? Tell me if you have any doubt.
So could you please repeat where PS system 2 is?
Right. Look, like this one, these part, let me show you with some different color. Suppose that these part where they are contact with another thylakoid, right? Another thylakoid. These part are called appraised part. Appraised part, right? So appraised part of thylakoid. These appraised part of thylakoid, they have photosystem second, PS second, and PS second is responsible for photolysis of water. And these part, which are the outside, they are in contact with the stroma, right? These part are called, these areas are called non-appraised part. Non-appraised part. The non-appraised part have photosystem. Is that clear?
Yes, sir.
Any more query? So this was the basic framework which you need to understand. Then only you will be able to, where the Krebs cycle goes on, where the Kelvin cycle goes on, where the C4 cycle goes on, and all that. Right?
What is lumen?
Arpit, be a bit loud. Yes.
But I'm saying, sir, what is lumen, sir, in this diagram?
What is
lumen? Lumen, sir.
lumen? Lumen is nothing actually. You know, I have this stylus, this pen. Is it visible? No, not visible. So,
Yes, sir. Yes.
okay. So inside the pen, there is a space. That space should be called lumen. So any structure, the space inside any structure is called lumen, right? It's a very generic term of biology. Means if it's
Okay, sir.
mitochondria, inside mitochondria there is a matrix, we will call that lumen, right?
Yes, sir.
Okay. So let's go to the next thing. Photosystem. What are the photosystems? Right? So where the photosystem are found, I have told you, photosystems are just the arrangement of us that you have to remember. This PPT you have to remember. The question I have been asked from here. This PPT. Look. What is the photosystem? Photosystem are just collection of some chlorophyll molecule or some pigment molecule. Better term is pigment molecule. Look. Photosystem have two parts. It's have two. This structure, this complete structure, have two parts. One part is a central or core part. Central or core part is called reaction center. Reaction center is that part from where the electron get excited, right? Reaction center is made up of a special type of chlorophyll A molecule. I have written over here. Reaction center. So this is the reaction center. It is made up of chlorophyll A molecule, and the name of chlorophyll molecule P700. That's why PS1 is also known as P700. Clear? Why we call it P700? Because the reaction center, the chlorophyll molecule, chlorophyll A molecule, which is present in the reaction center, is P700. Right? Now what happened? So a reaction center have a reaction center and a reaction center chlorophyll A molecule and a primary electron acceptor. So when it will take energy, it will take energy after the breakage of water molecule. It will accept electron, primary electron acceptor, right? And the energy will be provided by reaction center. Around the reaction center, there are some molecules, surrounding molecules. These are known as antenna pigment molecules. Antenna pigment molecules are made up of, so the number of antenna pigment molecule may be 250 to 400, right? These antenna pigment molecule are carotenoid and chlorophyll B molecule, right? These pigment capture the photon and finally they take the energy and transfer to the reaction. The reaction center give energy to the electron. That's how it works, right? And this complete structure is known as photosystem. So we have already discussed PS1 are found in non-appressed part. They have more chlorophyll A molecule, right? Chlorophyll B and carotenoid are found in less amount. This question I have been actually asked, that which of the following is wrong about PS1, which of the following is right about the PS1, that kind of the question. So it have more chlorophyll A molecule. Chlorophyll B and carotenoid are less. Photocenter is a dimer, means made up of two chlorophyll A molecule. So photocenter is a dimer of chlorophyll A, right? That is P700. It takes part in both. Let me write it on both there. It takes part in both cyclic and non-cyclic photophosphorylation, means cyclic and non-cyclic reaction. It takes part in both cyclic and non-cyclic photophosphorylation, right? What is the photophosphorylation? Formation of ATP. So formation of ATP is called photophosphorylation technically, right? Now what is the PS second? Come to the PS second. Both have the same structure. They are found in appressed part of thylakoid. Chlorophyll A, chlorophyll B, and carotenoid are found. Chlorophyll A and chlorophyll B are found in equal amount. Here both are found in equal amount, right? There the chlorophyll A was more. Here the chlorophyll A and chlorophyll B both are found in equal amount. Carotenoid are in higher side in PS1. Photocenter is a dimer of chlorophyll A molecule, means that is a P680. It have oxygen evolving complex, which help in evolution of oxygen. That is very important. It have oxygen evolving complex. Oxygen evolving complex is, in short, it is called OEC, oxygen evolving complex. What is the function of oxygen evolving complex? This oxygen evolving complex is responsible for breakage of water molecule, right? It is responsible for breakage of water molecule, 4H O2 plus 4 electron, right? And oxygen evolving complex break down the water molecule, or we can say it is responsible for photolysis of water, right? Photolysis of water. So what is the composition? This question I have been asked. What is the composition of OEC, oxygen evolving complex? Mn means manganese, calcium, and chloride is the part or component of oxygen evolving. Tell me, guys, is it clear or not? Is that
Yes,
clear?
sir.
Great. Now come to the non-cyclic photophosphorylation. Right? Electron transport chain or non-cyclic photophosphorylation. Here the electron is produced at one point and get consumed at another point. This is called Hill. It was discovered by Hill, right? So what is non-cyclic photophosphorylation? You have to remember only those components which I have encircled like this. So first what happened? There is a PS second. PS second, we know that this is found in oppressed part, right? And this is responsible for, it have oxygen evolving complex. It is written over here, OEC. It breaks down the water molecule, right? Water molecule means photolysis takes place. Water molecule is breakdown, right? Formation of oxygen and 4 electron takes place. These 4 electron goes to chlorophyll, a molecule 680, PS1. It provide energy, and that's why these jump and they become highly energetic. The electron get excited. These electron goes to a component called pheophytin, right? It is written over here. Pheo means pheophytin. Then from pheophytin, they reaches to PQ, plastoquinine. Plastoquinine, I have written over here. When they reaches to plastoquinine, after that they reaches to cytochrome B6F. Cyto B6F means cytochrome B6F complex. When they pass, these electron passes through, or and try to understand, when they are passing through these component, their electron is losing its energy. So when electron will pass through cytochrome B6F, what will happen? ATP will convert, ADP will convert into ATP. Means formation of energy is taking place. From plastoquinine, from cytochrome B6F, these electron passes to cytochrome F. From cytochrome F to plastocyanin, PC, plastocyanin. From plastocyanin, it comes to chlorophyll, P700, means PS1. Now electron have lose its energy a lot. Now what will happen? Again, P700 will provide energy to electron. Electron will again jump. Electron will get excited. It will go to A3, and from A3 it go to FX, and from FX it goes to FD, right? So this FX, that is the FES, ferrous sulfur complex, right? FES. Then it reaches to ferredoxin, FD. I have written over here. When it reaches to FD, the electron is finally accepted by. So who's the electron acceptor? Finally, electron is accepted by NADP plus and converted into NADPH. And NADPH is a form of energy. I have written over here 1 GTP is equal to 1 ATP, means GTP and ATP are equal. 1 NADPH is equal to 3 ATP, and 1 FADH is equal to 2 ATP. That also you have to remember. Go through with it and tell me, is it clear or not? Non-cyclic photophosphorylation.
Clear, sir.
Great. Come to the cyclic photophosphorylation. In cyclic photophosphorylation, electron comes back from where it has started its journey, right? So electron comes back to P700, right? Electron comes back to P700. Sorry, electron starts its journey from P700 and it comes back to it again here. So that's why this is called cyclic. When cyclic photophosphorylation will occur, that was the question and it has been asked. And that is very important. It will occur in low light intensity when the intensity wavelength is longer than 680 nanometer. Very important question. When carbon dioxide fixation is inhibited, means stop. When NADPH is not required. And in this cyclic photophosphorylation, only PS1 is involved, right? And one more thing, less energy production takes place. Less energy produced, right? From P700, electron it start its journey. It reaches to primary electron acceptor, reaches to ferredoxin. From ferredoxin, it reaches to plastoquinine. When from ferredoxin, it must go to NADPH, NADP plus. But NADP plus is not present. Then from ferredoxin, it will come back to plastoquinine. Plastoquinine, so when it will come from ferredoxin to plastoquinine, formation of ATP will take place, means formation of energy will take place. From plastoquinine, when it will come to cytochrome B6F, cytochrome B6F, where it will reach to cytochrome B6F, what will happen? Formation of ATP will take place. From cyto B6F, it will come to plastocyanin. Plastocyanin, so again it will come to P7. Right. So this is cyclic photophosphorylation, where ATP are produced, but with lesser. Electron again come back to P7. Clear, guys? Iram, are you getting everything? Heather?
Yes, sir.
Good.
Yes, sir.
Very good. chemoosmotic theory. So I have already told you that it have been asked. I have written over here who given this theory, and again it can be asked. Michel was the scientist in 1961. Michel given this theory. His name is Michel. Chemoosmotic theory was given by Michel in 1961. Photophosphorylation means ATP synthesis. Look, this is the layer which is visible in front of you. This is a thylakoid. Thylakoid, it's a structure of thylakoid. Now, what is happening, right? This thylakoid membrane. This is the thylakoid space. Water molecule breaks down here in PS second. Water oxygen will be released and 2 hydrogen ion will be released. So number of hydrogen ion is increasing inside the lumen, means inside the thylakoid. And this is the outside of the thylakoid, that is the stroma. Electron will reach to PS second to plastocunine, and plastocunine to cytochrome B6F. When electron will reach to pass through the cytochrome B6F, 2 hydrogen ion will be pumped in from the outer space to the lumen of thylakoid. Again, the hydrogen ion concentration is increasing inside thylakoid. From electron from cytochrome B6F goes to plastocyanin. From plastocyanin, they will reach to the photosystem 1. From photosystem 1 to ferredoxin, ferredoxin to NADP+, and from NADP+, the electron will be finally accepted and formation of NADPH will be taking place. And this NADPH will be utilized in Calvin cycle, means fixation of carbon dioxide. Again, now what will happen because of the passage of electron through these cytochromes? And all these cytochromes are found in the membrane of thylakoid, right? Due to passage of electron, hydrogen ions are more hydrogen ions are pumped in the lumen of thylakoid. Now what will happen? There is a concentration gradient, concentration difference between the lumen and outside of the stroma. So stroma will have low hydrogen ion concentration and thylakoid space will have high hydrogen ion concentration. What will happen? Hydrogen ion will always move from higher concentration to lower concentration. Hydrogen ion cannot go out from anywhere. There is only single space from where they can leak out. This space or this point is called ATP synthase enzyme. It's an enzyme, ATP synthase enzyme. So when the hydrogen ion will pass through the ATP synthase enzyme, what will happen? Formation of ATP will take place from ADP. At the passage of, at the leak out of every 2 hydrogen ion, 1 ATP will be produced. This theory, which explains how the formation of ATP takes place due to leakage of hydrogen ion, we call this theory chemoosmotic theory. And it was explained by Michel. How does the photophosphorylation or formation of ATP takes place? Tell me, is it clear or not? Clear?
Yes, sir.
Okay. So whatever we have studied, this was the light reaction. And light reaction goes on in the grain. Now come to the stroma. A stroma part where the dark reaction will go on. Dark reaction, why we call it dark reaction? Because it do not require the light. It goes on both day and night, right? Dark reaction is the process of fixation of carbon dioxide. Look, this is a Calvin cycle because discovered by Calvin Melvin. This is called C3 cycle because the first stable compound is 3 carbon compound. Look, this is the first stable compound, phosphoglycerate, PGA, and this is made up of 3 carbon. That's why it is also called C3. Right. Now, when this cycle will go one round, what will happen during this one round? One atom of carbon dioxide will be fixed, right? So, one carbon at one round. So, formation of glucose takes place from 6 carbon. how many rounds the cycle have to go to fix one glucose molecule? Every time it deliver one carbon atom. How many carbon atom are in glucose? 6 carbon atom. So when this cycle will go on one time, it will give one carbon atom to glucose formation, right? So how many times cycle have to go to fix one glucose molecule?
6 times.
Very good, 6 times, right? So the Kelvin cycle will go on 6 times, right? When 6 times Kelvin cycle will go on, then the 6 carbon atom will be fixed and formation of one glucose molecule will take place, right? Look, again, there is a substrate or compound. Name of this compound is RUBP, ribulose biphosphate. It is 5 carbon compound. This purple color circle represents the carbon and yellow color circle represents the phosphate. So RUBP, ribulose biphosphate. It is a 5 carbon compound and made up of 2 phosphate. What will happen? This RUBP will react with it will combine with carbon dioxide. In presence of enzyme rubisco, ribulose biphosphate carboxylase oxygenase. This is the name of this enzyme, rubisco. What is the peculiarity with the rubisco? What is the specialty of rubisco? That question I've been asked in medical NEET exams. What is the special with the rubisco? Anyone of you?
It is the most abundant enzyme.
Very good, excellent. Very good. This is the, if you want, you can note it down. This is the most abundant enzyme on the earth. First thing, this enzyme is made up of protein. So another thing is that this is the most abundant enzyme of the earth. This is the most abundant protein of the earth. Both are the correct statement, right? So rubisco is the most abundant enzyme on the earth. I have one more question. Which is the most abundant organic matter of the earth? Most abundant organic matter of the earth. Guess, try to guess that. Organic matter means, let me give you hints. It's a sugar. The most abundant organic matter on the earth. Come on. All the plant are made up of a polysaccharide. What is that thing? Oh. Cellulose. All the plants are made up of cellulose. It's a sugar. Means it's organic matter. So the most abundant organic matter on the earth is cellulose. The most abundant protein on the earth is rubisco. The most abundant enzyme on the earth is rubisco. Okay. The name of rubisco is ribulose biphosphate carboxylase oxidase. In the last, there is a suffix, that is the carboxylase and oxidase. Why? Because if the carbon dioxide concentration will be more, it will react with the CO2. If the oxygen concentration will be more, it will react with the O2. So it can react with the both CO2 as well as O2, right? CO2, look, now what happened? CO2 react with RUBP in presence of rubisco. Formation of 2 molecule of a 3 carbon compound 2 phosphoglycerate takes place. This 2 phosphoglycerate means 2 PGA at the expense of 2 ATP, 2 NADPH, right? That means reduction takes place. So formation of 2 molecule of a 3 carbon compound, that is glyceraldehyde 3 phosphate G3P will take place. Now there are total 6, 2, 3 just 6 molecules, right? So now there are total 6 molecules, 6 atoms. So out of these 6 atoms, 1 atom is given, 1 molecule or 1 atom of carbon is given for the glucose formation, right? And rest, so how many there are left? There are total 6 carbon, 1 carbon is given, so there are left only 5 carbon, 5 carbon and 1 phosphate. So formation of ribulose phosphate will take place. Again, ATP will be spent and formation of ribulose biphosphate takes place. The first step is called carboxylation because addition of carbon dioxide takes place. Second step is called reduction. because it get reduced, and the third part is called regeneration because the regeneration of ribulose biphosphate takes place. Yes, Anna have told right correct answer. Cellulose the most. I forget go into inbox while teaching. Yeah, I'm sorry for that. So is that clear, or if you want to any more explanation? If you want any more explanation, you are most welcome. I'll tell you. Tell me. Go through with it. If you have any doubt, I'll tell you. But you have to remember. You have to remember this cycle. This at least this cycle. Marita, okay?
Yes, sir.
Alia, are you getting everything? Alia, Samaira,
Yes, sir,
all
clear.
clear? Very good. Fine. Again, I have explained the same thing in the form of regeneration. This one, carboxylation RUBP plus CO2 means formation of 2-PGA. Reduction 2-PGA plus 2-NADPH formation of G3P and NADP. Same thing, whatever we explained over here. Now, what is the photorespiration? Photorespiration is a bad phenomenon which reduces. This is also known as C2 cycle. Photorespiration is a bad phenomena, and in this, the loss of photosynthesis takes place. Means the loss of uh, food production takes place, right? Glucose production takes place. Loss of glucose production takes place. Why and how? Let me tell you. I have already told you that RUBP combined with CO2 in presence of Rubisco and form 2-PGA. Normally, in normal CO2 concentration is normal. If in normal CO2 concentration, C3 cycle will go on. But I have already told you that Rubisco is capable of reacting with oxygen. When oxygen concentration increases, what will happen? This RUBP will react with O2 in presence of Rubisco. Formation of one PGA, one phosphoglycine, but formation of a phosphoglycolate will take place and loss of one carbon atom will take place. So previously in C3 cycle we have discussed formation of 2 PGA takes place, right? So this is the 3 carbon compound, but here formation of 2 PGA molecule is not taking place. One PGA molecule is formed and one phosphoglycolate is formed. PGA is 3 carbon compound, but phosphoglycolate is 1 carbon compound. What will happen? One carbon atom will be lost. That's why productivity reduces. So what happened with this phosphoglycolate? Try to understand. This is called C2 cycle. Why? Because the first stable compound is phosphoglycolate, that you have to remember. And phosphoglycolate, it is 2 carbon. Carbon dioxide, 3 PG, phosphoglycolate. This phosphoglycolate, look, ribulose biphosphate, it react RUBP, react with O2. Formation of phosphoglycolate takes place, it is 2 carbon compound. Now what will happen? This 2 carbon compound from chloroplast, a phosphoglycolate, will move to another organelle which is called peroxisome. It will convert to glycolate. Then glycolate will convert to glyoxylate, then glycine, and glycine will move to the mitochondria. In mitochondria, when glycine will move to the mitochondria, what will happen? Glyoxine will remove one carbon dioxide atom. Again, one carbon dioxide atom will removed, right? Convert into serine. Serine will convert into glycerate. Glycerate will convert into, again, glycerate will go to this one. Here, again, chloroplast and convert into phosphoglycolate, and formation of ATP means expense of ATP takes place. So first thing, this is energy consuming process, right? Very important thing, this is energy consuming process. 25% of productivity reduces. 25% of productivity reduces because of this phenomenon. When oxygen concentration is, that is also important. When oxygen concentration is 2 to 3% to 21%, right? Means it's 2 to 3% more to 21%, right? Formation of phosphoglycolate takes place. Only one molecule of PGA is produced. It releases CO2. So if this phenomena is taking place, then cell organelle like chloroplast and mitochondria will eliminate or will release the carbon dioxide, and 2 ATP are spent along with that. So it is energy consuming process as well. It wastes the energy along with the productivity reduces. So this process called photooxidation. This photooxidation takes place in C3 plant, not in C4 plant. Tell me, is it clear or not? Okay, fine. So what here you have to remember, the question that how much productivity reduces, what should be the more concentration from 21%, what should be the more concentration of oxygen, right? Which is the first compound, and what is formed as a first compound is phosphoglycolate and 2-PGA. Crange anatomy, very important point. What is crange anatomy? Now, crange anatomy is found in C4 plant. This is evolution. So, as per my view, the C4 plant, what I consider C4 plant are more advanced than C3 plant. Why? Because in C4 plant, they have developed that mechanism so that the photo oxidation or photo respiration do not take place, right? Means RUBP do not combine with the O2. Look, first, what is the crange anatomy? In the crange anatomy, there is a vascular bundle in the leaves. Around the vascular bundle, there are some cells which are called bundle sheath cells. Bundle sheath cells have chloroplast, which are larger in size and no grana is present. Means they are called agranal. That is important. That is a question. Bundle sheath cells have chloroplast. They are different chloroplast. They are larger in size and they do not have grana, means they are agranal. Again, around the bundle sheath cells, there are mesophyll cells. Mesophyll cells have a chloroplast which are smaller and they have developed grana, right? They have developed grana. So there are two kinds of the chloroplast. This is called dimorphic chloroplast. So where dimorphic chloroplasts are found? Dimorphic chloroplasts are found where the crange anatomy is present or in C4 plant, right? What are dimorphic chloroplasts? The chloroplast in the bundle sheath cell do not have grana, agranal, and the chloroplast in the mesophyll cell, they have grana, they are granal. So there are two types of the chloroplast, that is called dimorphic chloroplast. Again, this is mesophyll cell. One more thing which I want to tell you, this bundle sheath cell is impermeable to oxygen. So let me recall you. Actually, Rubisco can react with the oxygen, but Rubisco is found in the bundle sheath cell where oxygen cannot go. That's why photorespiration can never take place in this crange anatomy or C4 plant. Look, carbon dioxide comes in mesophyll cell. There is an enzyme PP carboxylase, phosphoenol carboxylase, phosphoenol pyruvate carboxylase. PP carboxylase, it combined with carbon dioxide. PP is 3 carbon compound. And when it combined with one more carbon compound, it makes 4 carbon compound. So first stable compound is glyoxylate. OAA. First carbon compound is oxaloacetic acid or oxaloacetate. So that's why this is first carbon compound and this is 4 carbon compound. That's why this cycle is called C4 cycle. Oxaloacetate convert into malate. Malate move into bundle sheath cell from plasmodesmata. In bundle sheath shell, malloid break down into pyruvate, that is 3-carbon compound, and a carbon dioxide is released. This carbon dioxide is fixed by Rubisco, and C3 cycle goes on here, right? And sugar formation takes place. Pyruvate will come back. Again, ATP will be expelled, and pyruvate will convert into PEP, right? So, what is that? What is the benefit of craniotomy? In craniotomy, what you have to remember, dimorphic chloroplasts are found. Photophosphorylation cannot take photorespiration cannot take place. Why? Where the Rubisco is present, that cell is a bundle sheath cell which is impermeable to oxygen, and dimorphic, and they consume more energy than C3 plant, right? So they require, because here ATP is spent, right? So this is the craniotomy and C4 cycle. Go through with it and tell me is it clear or not. Is that okay? Done? No doubt, no questions. It is very important. It's more important than C3 cycle. C4 cycle, the same thing which we discussed, C4 cycle. So craniotomy, yeah, what is the more important? Where C4, which are the C4 plant and where the craniotomy is found? All the example have been asked. All these maize, C4 plant or craniotomy is found in maize, sugarcane, sorghum, and panicum, right? So remember all 4 examples. All have been asked. That question have been asked, right? First compound, oxaloacetic acid, OA. The rest of the thing we have discussed, already discussed. You have to remember all these names. All they have been asked. Maize, sugarcane, sorghum, and panicum. They all have the craniotomy and C4 cycle goals. They are the C4 plant. They are more advanced than C3 plant. What are the CAM plant? CAM plant are also known as succulent plant. For example, cactus, Apansia. Apansia, that cactus, you must have seen this kind of the diagram, this kind of the plant. This kind of the desert plant, it have some spines like this way. Yeah. This kind of the plant, right? This is Apansia basically, and they have some spines. Basically, leaves get modified into spines, and these stem are green and they perform the photosynthesis. And this is the stem which perform the photosynthesis. The stem, what we call this stem, basically stem is green. The stem is green, and this is known as phylloclad. This stem, the name of the stem is phylloclad, and they perform photosynthesis. So basically, these stem, they have inside the stem there is a mucus, some sticky substance, mucus, right? This mucus hold water. This mucus hold water, right? That's why, and this mucus plus this system is spongy. So they hold water, they absorb water. That's why these kind of the plant are known as the succulent, right? So Apansia and sedum, right? Kalanchi, right? Kalanchoe, Apansia, sedum, Kalanchoe, they all are succulent plant, right? They have, so what they do, they have crassulacean acid metabolism. They are same as C4 plant, right? These succulent plants are same as C4 plant, means C4 cycle goes on. They are found in desert and arid region. In daytime, what happened? In daytime, they do not open their stomata and photosynthesis goes on, right? In nighttime, they open their stomata and absorb the oxygen, absorb the carbon dioxide, right? And open stomata at night, right? And PEP in day, light reaction perform, right? PEP perform the light reaction in day, and Rubisco perform its reaction in the night, and dark reaction takes place in the night. So daytime in succulent plant or CAM plant, respiration acid metabolism plant. So what they do in the night, they take the carbon dioxide and they fix this carbon dioxide in the form of respiration acid. In daytime, CAM respiration acid break down and carbon dioxide is released. And in day, they perform the light reaction. So light reaction goes on in day and dark reaction in night. So these are plant are called CAM plant. Clear? Remember succulent plant, remember CAM plant. Samira, Marita, clear? Heather, Iram,
Yes, sir.
Yes, sir.
Alia, Arpit.
Yes, sir.
So last but not the least, the factor affecting the photosynthesis. So it's very simple. Law of limiting factor was given by Blackman. An important question. It has been asked. Blackman says the factor which is present in the least amount. The factor which is present in the least amount. The factor which is present in the least amount will decide the rate of reaction. Right? It was given by the Blackman in 1904. Right? So CO2 concentration, it goes up to 0.5. Right? If goes up to 0.5, photosynthesis will increase. So normally, what is the normal concentration of CO2? So normal concentration of CO2 is 0.03, right? If it will go up to 0.05, then what will happen? Photosynthesis will increase. Beyond it, the photosynthesis will not increase, right? And for the C4 plant, the concentration of CO2 will be 360 microliter per liter. In C3 plant, 450 microliter per liter, right? If light or temperature increases, photosynthesis will increase up to certain limit, and after that it will become constant, it will not increase. And beyond a particular light, beyond a particular limitation of light, the cell organelle will start dying. Water, if water is less, then photosynthesis rate will decrease. If water is more, then photosynthesis rate will be better, right? So these are the factors. You have to remember only the carbon dioxide concentration part, right? That is the point. And law of limiting factor was given by Blackman. Is that clear to all of you?
Yes,
So,
sir.
very good. That's it for the day. And we have completed the photosynthesis. Again, go through with PPT. Note down if you have any doubt. Go through with NCRT and last 5-year previous year question paper. Come with your doubt if you have doubt. In next class, we'll start with the next chapter, right? So bye-bye. Take care. See you in next class.
Thank you, sir.
Yeah, welcome.
Thank you, sir.
Sir, can I ask what did you cover after the ecology?
Be a bit loud. Actually, you're not.
Sir,
Yes.
what did we cover after the ecology part? Because I missed my yesterday's class.
Yeah, actually, ecology. When I finished the ecology, then I started with the transport and plant. Right, that was the first chapter which we started in plant physiology. Then we did the mineral nutrition. So today, in